Thank you for watching this presentation. Today I'm talking about the Admp functions in cortical development. Admp is known as a neuroprotective protein. Admp mutation is one of the most frequent genetic mutations causing autism spectrum disorder and intellectual disability. Admp patient shows multiple symptoms. These include seizure, defect in motor function, anxiety, and ADHD. This illustration shows the structure of Admp protein. There are two main functional domains, which are N-terminal and C-terminal domains. In N-terminal domains, there is the NAP domain, which binds to microtubule plus end binding protein EB1 and EB3 and regulates microtubule polymerization and assembly. In C-terminal region, Admp has a nuclear localization signal and DNA binding domain, indicating that Admp functions as a transcription factor. However, there are limited studies about the Admp functions during the neurodevelopment. Therefore, we analyzed the functions of Admp in cortical development. We used in vitro and in vivo methods. In vitro primary cortical neuron culture, we prepared cortical neurons at E15.5 and transfected with shRNA. After 48 hours, neurons were replated and cultured additional 48 hours. In vivo analysis, we used in utero extraporation with plasmids such as shRNA plasmid using E15.5 embryos and analyzed the neural morphology and neural connectivity and activity. We performed ex vivo live imaging at P0, neuromorphological analysis at P15, and neural connectivity and neural activity at P30. During cortical development, postmitotic neurons migrate and reach the cortical brain. Then, neurons start neurite formation. Neurite formation can be classified into two steps: Neuralite Initiation and Neuralite Elongation outgrowth. In vitro primary neuron culture, round-shaped neurons start extending philopodia and lamellipodia and then create primitive neurites. This step is called Neuralite Initiation. Then microtubules invade into primitive neurites and primitive neurites start extending. This is the elongation step. In our study, we focused on the Admp functions in neuronal morphology, especially Neuralite initiation and elongation. In vitro cortical neuron culture, we knocked down Admp by shRNA and analyzed the neurite number from SOMA and neurite length. Neurite number relies on neurite initiation. In contrast, neurite length relies on neurite elongation. Admp deficient neurons showed increase in neurite number and neurite length in the longest neurite, which is likely become the axon. Next, we analyzed Admp functions in vivo using in vitro extraporation. We targeted layer 2-3 pyramidal neuron in the somatosensory region. At P15, neurons extend the axon, the apical dendrite, and basal dendrites. We first analyzed the dendrite morphology. We found that Admp knockdown neurons extended the apical dendrite, but its angle was broader as compared to the control neuron, which extended the apical dendrite straight to the pial surface. We also found that Admp knockdown resulted in increase in basal dendrite number and decrease in basal dendrite length. We also analyzed if Admp knockdown affects axon length. We used the same P15 samples. In control neurons, the axon extended and passed the midline, but at P15, the axon projection did not invade into another side of the cortex. In contrast, the axon bundles of Admp knockdown neurons reached and invaded into another side of the cortex. We measured the axon length from the midline and found that the longer axon length in Admp deficient neurons. These data indicate that Admp regulates Neuralite formation, including Neuralite initiation and elongation. To analyze the effect of Admp deficiency on Neuralite initiation, we performed ex vivo live imaging. We did in vitro electroporation at E15.5 and performed live imaging on brain slices at P0 when the postmytic neurons have just reached the cortical plate and start Neuralite initiation there. We found two major defects in Admp deficient neurons. First, Admp deficient neurons are less active in terms of Neuralite dynamics. Second, Admp knockdown neurons have thicker neuralites. These are videos from the about 24-hour recording. In control neurons, round-shaped neurons start extending some neuralites, and these neuralites are active by repeating extending and shrinking. In contrast, Admp neurons had already one single neuralite at the beginning of the decoding, but not so active, rather stable without major morphological change. We measured neuralite length at the starting time point, neuralite growth, velocity in growth and shrinking. We found that Admp deficient neurons had the neuralite, but less active and stable in neuralite dynamics. Also, we noticed that these neuralites in Admp deficient neuron is significantly thicker by measuring the width of the neuralite. These data indicate that Admp is a key player in Neuralite formation, both in Neuralite initiation and elongation. Also, our data suggests that different functions in the axon, the apical dendrite, and the basal dendrites. Our next interest is whether Admp deficiency affects to the spine formation and morphology. Using P15 samples, we analyzed the spine density and morphology, which relates to maturity. Admp deficient neurons had less spines. The control neurons had more mature type spines, such as mushroom shape. But Admp deficient neurons had more immature spines, such as stubby and thin shapes. This data suggests that Admp deficient neurons could receive less inputs. To analyze the neural connectivity, we adopted the state-of-the-art technique called graphic. The graphic is based on the reconstitution of two fragments of GFP protein, N-terminal and C-terminal fragments. When each portion is expressed in two different cells and they closely contact each other, the full-length and functional GFP is reconstituted by two GFP fragments. In neurons, only synaptic gap is close enough to reconstitute GFP, so we can visualize the neural connections in synapses. We used in vitro extrapolation using two groups of plasmid and injected them into two cortical hemispheres. One includes N-graphic plasmid coding for N-terminal GFP and H2B M-cherry tag, plus shRNA plasmid with BFP tag. The other includes C-graphic coding for C-terminal GFP and M-cherry, plus shRNA with BFP tag. We injected C-graphic and shRNA into the right hemisphere and N-graphic and shRNA into the left hemisphere. M-cherry and/or BFP can visualize the dendritic morphology, including spines. So we counted the GFP puncture on M-cherry positive dendrites and spines. Admp knockdown resulted in increase in GFP puncture number as well as increase in puncture in the shaft region. Our experiment targets excitatory pyramidal neurons and analyzed the intracortical cortical connection. Our results indicate that Admp deficient neurons receive more excitatory input even in shaft region of dendrites. Our data indicate that Admp neurons have longer axon and more basal dendrites, suggesting that they could receive more inputs. On the other hand, they have shorter basal dendrites and less spines and more immature spines, suggesting that they could receive less inputs. To test if Admp deficient neurons are hyperactive, we performed ex vivo calcium imaging. We used P30 brain slices electroporated with GCAMP6 calcium indicator and shRNA. Admp deficient neurons showed hyperactivity in spontaneous condition compared to the control neurons. In the condition we use here, more than 23% change of fluorescence intensity indicate an action potential. We found that more number of Admp deficient neurons showed spontaneous action potential. Our series of experiments clarified the multiple functions of Admp in neurodevelopment, including in neurite formation, spine formation, neural activity, and connectivity. And this suggests that Admp is a key player in neuronal development. Another interest about Admp functions is how the functions of Admp as a transcription factor and a microtubule regulator are regulated. It is known that these functional changes are related to neuronal differentiation. Also, the Admp localization changes from the nucleus to the cytoplasm during neuronal differentiation. In immature neurons, Admp is distributed in the nucleus. In mature neurons, Admp is distributed in the cytoplasm. We have been working on the functions of 1433 proteins in neurodevelopment, and one of the major functions of 1433 is to shatter the binding target from the nucleus into the cytoplasm. Therefore, we tested if 1433 regulates the Admp distribution by using 1433 inhibitor. In immature neurons, in neurosphere created from mouse cortical neural progenitor cells, Admp is predominantly localized inside the nucleus. In mature cortical neurons, Admp is localized in the cytoplasm and prenuclear region. In mature neurons, we transfected the plasmid codeine diphopain, peptide 1433 inhibitor, and analyzed the Admp distribution. Admp was trapped inside the nucleus, suggesting that 1433 binds to the Admp and shatters it into the cytoplasm. And also, by using pull-down analysis, we confirmed that it's 1433 epsilon, one of the 1433 family protein isoform, binds to Admp. Finally, I thank my team and collaborators. This project was mainly conducted by my graduate student, Sadie, in support with other lab members. We also thank Dr. Simogori and her lab member, who developed the graphic technique and kindly agreed to share graphic plasmids. Also, these experiments are supported by the Drexel University and our department, Neurobiology and Anatomy, and the funding from NINDX. Thank you for your attention. If you have any questions and comments, please feel free to email me. I'm happy to discuss via email or even a Zoom meeting. Also, if you want to know Admp project in more detail, please check out the preprint we recently posted. Finally, if you are interested in other projects happening in my lab, please check out the link shown here. Thank you for watching the presentation.